Well, thank you for that introduction. I'm very pleased to be here. It was a pleasure to be asked. I'm slightly daunted at having to follow the previous plenary lectures, but I will do my best. The title of the talk was Vagal Neurocircuits and Neurogastroenterology, and I said to Niall, this is a really broad topic. Is there anything in particular you would like me to talk about? And he said, oh, no, it's up to you. I thought, well, be careful what you wish for. But I decided I was going to kind of restrict things a little bit because I didn't necessarily think that anyone would want to sit and listen to an hour of electrophysiology that I've been doing. So we'll introduce some other topics. What I'd like to do is actually give a brief, well, relatively brief, background overview of vagal neurocircuits. And in part that's because while I think the vagus is the most important nerve, I understand that not everybody lives in the same vagal-centred world that I do. And so then I thought, well, I'll pick a few areas of interest within vagal neurocircuits in terms of things that have been happening recently. And I picked 3 because the best sermons have 3 parts. And so I thought I'll talk a little bit about the vagal anti-inflammatory reflex, or the cholinergic anti-inflammatory reflex. This is not an area that I have worked on. I don't have any plans to work in this area in future, but it's a huge area of interest within the field. And within the past 18 months to 2 years, there have been several papers which have come out which have shed new light on what the vagus does and doesn't do in terms of this cholinergic anti-inflammatory reflex. I'd then like to talk a little bit about vagal neurocircuits and neurological disorders. That, again, is becoming a hot area of interest, as was outlined yesterday in several talks and in many of the posters, the potential role of the vagus and vagal neurocircuits in Parkinson's disease. And then my indulgence is talking a little bit about developmental programming of vagal neurocircuits because that's what I have been working on recently. But I think this may have profound consequences in terms of lifelong outcomes and responsivity in both rodents and human models. So this is the vagus nerve, highlighted in yellow. It's a mixed sensory motor nerve. Anywhere between 65 to 85% sensory, the remainder being motor. So it's predominantly a sensory nerve, and many people forget that. The sensory cell bodies are contained within the paired nodose ganglia, or the nodose ganglion complex. This is located in the periphery at the height of the transverse process of the first cervical vertebra, so just outside the central nervous system, whereas the motor cell bodies are located within the dorsal aspect of the caudal brainstem. And as can be seen here, the vagus provides the parasympathetic innervation to almost the entire GI tract, from the pharynx and larynx up to the terminal 2/3 of the colon. Embryologically, the vagus appears to be present throughout the colon, but in adulthood, the vagal innervation appears to terminate somewhere around about the splenic flexure, with parasympathetic innervation to the descending colon being supplied by the sacral spinal cord. So we've heard several talks that have introduced these vagal afferents. The afferents are the sensory cells. And it's clear that, as in a great many areas of neuroscience, the structure and the location dictates their function. So we have, in terms of responses to muscle, to stretch or distension of the intestine, we have the intraganglionic afferents, which form intraganglionic lamellar endings, mostly within the myenteric ganglia. And we have these intramuscular afferents, which terminate within the longitudinal and or the circular muscle. But we also have chemosensitive afferents here, which penetrate up to the tip of the mucosa, or these muscular mucosal afferents, which appear to be a hybrid of chemosensing and a little bit of tension sensing too. So these early studies, these beautiful anatomical studies, carried out by, in this instance, Ed Fox and Terry Powley, and by Bob Phillips and Terry Powley, show the density of this afferent innervation. And here, these IGLEs can be seen. This is the vagal nerve trunk coming down the esophagus and entering into the stomach, and can be seen arborizing into almost every myenteric ganglion. These are thought to be tension mechanoreceptors that sense muscle distension or contraction through stretch-operated ion channels, whereas the intramuscular arrays can be seen to be aligned in series with the muscle, and they might be tension or length receptors, although the structure and the function of these is less well defined. But what is important is that the sensitivity, the mechanical sensitivity of these afferents is not static. It's modulated by several circulating factors, including neurohormones released by enteroendocrine cells, such as leptin or ghrelin. They also have a very profound circadian rhythmicity to them. So in a series of very rigorous experiments from Amanda Page's lab, they have shown that the sensitivity of these IGLEs, these mechanosensory afferents, decreases at night. And these are in rodent studies, and that's important because rodents are nocturnal, so they eat more during the night. And this decreased sensitivity of the afferents allows them to ingest more before they perceive that tension or stretch, whereas during the day, these afferents become much more sensitive to stretch or distension, so the rodents eat less during the day. But they also know that exposure to a high-fat diet ablates that circadian rhythmicity, and rodents eat more during the day, eat larger meal sizes, put on weight, and become obese. And a potentially intriguing translational aspect to this is the fact that this circadian rhythm is lost relatively quickly if you switch the light cycle. So within 3 to 4 days, switching the light cycle, that circadian rhythm is lost. And that may be very important for shift workers who can have quite profound gastrointestinal disturbances and an increased incidence of obesity. So dye studies, these original studies identifying these IGLEs were done in this beautiful example from Terry Powley by dye injections. They'd inject neuronal tracers into the nodose ganglion and then examine those endings within the myenteric plexus. And here we can see this one afferent fibre entering the myenteric ganglion and arborizing within the myenteric plexus and provides this very dense innervation. These studies were necessarily non-selective. It was a dye injection. These were lipophilic dyes that were transported through the vagus nerve, so we didn't have any further details about any specificity of these afferents. But newer, more recent studies taking advantage of the genetic techniques that are now available have given us some idea, potentially, about phenotypic characteristics of these afferents. So in this study, from Stephen Liberley's lab, they have this GLP-1 receptor mouse, and by injecting this AAV TD tomato, they can identify these GLP-1 receptor-containing afferents which enter the stomach. And as with all these vagal afferents, they enter from the minor curvature of the stomach and spread around the major curvature. But we can clearly see these arborizations, and in higher magnification, it's clear that these are IGLEs. So these GLP-1 receptor-containing afferents appear to be mechanosensitive. But what was interesting to me was how stretch-sensitive vagal afferents appear to be exactly the same regardless of the organ that they're sensing stretch in. So these are GLP-1 receptor-containing afferents in the stomach. This study from the same group looked at stretch-sensitive vagal afferents in the lung. And at higher magnification, you can see these form exactly the same arborization in terminal fields as they do in the stomach. And in this study, reviewed by Mark Chaplot, they're looking at vagal afferents which terminate in the aortic arch. So these are baroreceptor-sensitive afferents. So what they all have in common is that they sense stretch. But even this presents potential problems and a caveat in identification or characterization of these afferents, because these were identified with TRPC ion channels. These are also present in gastric or gastrointestinal vagal afferents. And these ones in the lungs were identified as containing the P2 receptor, which is also present in gastrointestinal vagal afferents. So this leads to the potential that what is actually defining these afferents is anatomy and development, because there is no real reason for a mechanosensitive afferent to be different. All it needs to do is respond to stretch in whichever organ it's innervating. But we also have these chemosensitive afferents. And again, with dye studies carried out by Terry Powley, we see this villus afferent, this single afferent fibre entering one villus and arborizing. These will clearly be in a prime location to respond to luminal factors. But we also have some afferents which surround crypt cells, and these are quite clearly going to be well positioned to respond to enteroendocrine hormone release. But again, non-specific, we have no further details as to the phenotype of these neurons. But in this same study, that demonstrated that the GLP-1 receptor-containing afferents were mechanosensitive. We know that GLP-1 receptors don't appear to be chemosensitive. They are not innervating the villi. But GPR65-containing afferents are. This is an orphan receptor. They are not sensitive to CCK or GLP-1, but they are sensitive to 5-HT or serotonin, and they are activated by luminal contents following food ingestion. So these do appear to be chemosensitive afferents. So the cell bodies of all of these afferents, regardless of whether they're gastrointestinal, respiratory, or cardiovascular, are located within the nodose ganglion or this nodose jugular ganglion complex. Whether or not there's a distinct jugular petrosal ganglion appears to be species dependent. In the rat, which I normally work in, there really isn't any distinction. It's very much one amorphous blob. And it has long been a question within the field of, is there any viscerotopic organization within this ganglion? And studies from Stephen Altschuler suggest, well, there might be. There may be a very broad, very loose viscerotopic organization, because in dye labeling studies, he showed that afferents which innervate sort of proximally located organs tend to be located in the rostral poles of the ganglia, whereas afferents which innervate more caudally located organs tend to be located in the caudal pole. But it's clearly a very loose organization. And this is only highlighting afferents from sort of the upper GI tract, which means that within these ganglia, there also must be afferents or cell bodies of afferents which innervate other organs. This close proximity of afferents may actually be very important from an autonomic homeostatic point of view. There is a real reason that your stomach should know what your lungs and your heart and your pancreas are doing. So by being closely opposed to each other, it allows for the possibility of crosstalk. And crosstalk within vagal afferent neurons was demonstrated in a beautifully elegant study from Danny Weinreich's lab. What they did was they took out the nodose ganglion and attached nerves, including the vagus nerve proper as well as the recurrent laryngeal nerve. What they were able to do was record from a vagal afferent neuron within the nodose ganglion, and by stimulating the recurrent laryngeal nerve or the vagus nerve caudal to the point at which the recurrent laryngeal nerve split off, they were able to antidromically activate this neuron and so tell which nerve branch that afferent neuron was projecting through. And what they found is that neurons that project through the recurrent laryngeal nerve were actually excited, were depolarized following stimulation of the vagus nerve. So there was cross-depolarization. This appeared to occur through both somatic release of the neurotransmitter, which was a little bit heretical, that the cell body can itself release neurotransmitter, but it clearly appears to occur here, as well as a generalized depolarization resulting from alterations in the extracellular ion concentration. So this means that there may be a means by which vagal afferents, sensory systems, can respond to stimulations of other organs. And that's good in a physiological sense. It may be bad in a pathophysiological sense. And certainly it's long been known that there's an increased tight correlation between the incidence of chronic cough and gastroesophageal reflux disease, as well as airway hyperresponsiveness and IBS. The neurocircuitry involved in the esophageal bronchial reflex has not yet been defined either in humans or in animal models, but this type of cross-depolarization and crosstalk at both peripheral and central levels may play some role in explaining these very common comorbidities. So the central terminals of these vagal afferents then enter the brainstem, and here they terminate within the nucleus of the tractus solitarius. And here there does appear to be a slightly tighter viscerotopic organization in terms of the termination of these afferents. So this is a horizontal section of a brainstem. And in this study from Stephen Altschuler, you can see that the terminations of afferents innervating the esophagus are located much more laterally than the ones that innervate the stomach. And these differences can be seen much more clearly if we look at a coronal section, so a transverse section through the brainstem, where we can see these sort of subnuclei. These are artificially imposed areas of interest. You cannot tell just looking at the brainstem what particular area you would be in. But we know that esophageal afferents tend to terminate within the centralis subnucleus, whereas gastric afferents terminate within the gelatinosus and the medialis. However, again, there has to be some crosstalk here, some idea of cross-depolarization or cross-excitation, because cardiorespiratory afferents also terminate within the medialis NTS, respiratory particularly in a ventrolateral NTS. But what is clear is this idea of lines of specificity, which is very common within vagal afferent physiology across fields, that one type of afferent responds to a particular type of stimulus, enters the brainstem and synapses onto a particular NTS neuron, and that NTS neuron is devoted to that function, and that NTS neuron has a particular target itself. So we have pathways of specificity. And this idea of specialization of the termination of these afferents or a viscerotopic organization is underpinned again by these more recent genetic tracing studies. In this one from Laurent Gautran's lab, here he has MC4R receptor-containing vagal afferents. So these are afferents that are involved in satiety signaling. And here they enter the brainstem through the tractus solitarius and appear to terminate particularly within this subpostremal or commissuralis area. Whereas these studies looking at those GLP-1 receptor-containing afferents, so the mechanoreceptor afferents, seem to terminate within the brainstem here, probably in the medialis area, whereas the chemosensitive ones appear to terminate again in a very similar area, this subpostremal area. And there's no overlap. So again, this idea of specific lines of sensory information and sensory inputting into the NTS. So the NTS is organized, at least in terms of the areas that they receive afferent inputs from. What about the efferent neurons, the motor neurons? And the dorsal motor nucleus of the vagus appears to be organized in terms of the branch of the efferent vagus nerve that they send their projections through. So in rodents, there are 5 vagal branches. In humans, there are 9, but certainly in the rodent studies, what we can see is that the left cervical vagus, the left vagus nerve, crosses and innervates the ventral or anterior stomach, as well as having branches of what would be the hepatic branch, which would innervate the intestine and the liver, and the accessory celiac branch, which is going to innervate the intestine, whereas the right cervical vagus innervates the posterior stomach, and the celiac branch proper will then innervate the intestine as well. So what this then suggests is that damage to discrete vagal branches may actually exert organ-specific effects, just as damage to particular areas of the dorsal motor nucleus of the vagus may have organ-specific outcomes. Gastric projecting vagal motor neurons tend to be located more medially, whereas those that innervate the intestine are located in the lateral tips or the lateral wings of the dorsal motor nucleus of the vagus. So these don't seem to be organised in the same way as the NTS. These are organised in terms of the way the efferent branches that they go through. The efferent distribution of these vagal motor neurons is incredibly dense in the proximal stomach, proximal and upper small intestine. This early study from Hans-Rudi Berthoud, they had labelled all of the myenteric neurons using a fluorogold, an intraperitoneal fluorogold injection, which just labels all neurons. This is the same field of view, illustrating the vagal efferent fibres that were labelled following a dye injection in the dorsal motor nucleus of the vagus. And at higher magnification, every neuron is seen to be contacted by these vagal efferents. And this density of innervation is confirmed by the neurogenetic labelling techniques. Again, from Laurent Gautron's lab, this is FOX2B containing vagal efferents, which are just labelling all of these visceral neurons. This is cholineacetyl transferase, so it's labelling all cholinergic neurons, and you can see how dense this innervation is. So the efferent vagus has an extremely powerful role in regulating the stomach and the upper GI tract, but this density of innervation declines as one progresses distally through the intestine, and by the time we get to the terminal colon, there's very less role, there's a significantly lesser role of the vagus in controlling that function. So this is our typical vagal-vagal reflex scheme. Afferent terminals within the gastrointestinal tract are going to respond to peripheral sensations. Their job is to transduce those sensations into an electrical signal and relay that signal centrally. They'll enter the brainstem through the tractus solitarius and terminate on neurons of the nucleus of the tractus solitarius. The NTS is a major integrating centre in the brain. So in addition to receiving this vast volume of sensory innervation, it also has reciprocal connections to several other brainstem and higher CNS areas involved in autonomic homeostatic control. They are going to integrate and assimilate all that information, and then, from the point of view of vagal-vagal reflexes, relay that assimilated information to the adjacent dorsal motor nucleus of the vagus. These are preganglionic parasympathetic motor neurons, so they're cholinergic. They are going to relay that signal back to the peripheral organ, in this case the stomach, through the efferent vagus nerve, releasing acetylcholine to activate nicotinic receptors. All of these neurons are cholinergic, but we know that they form at least 2 pathways. There is an excitatory cholinergic pathway, activation of which releases acetylcholine to activate muscarinic receptors, increasing gastric tone and motility, or there's a non-adrenergic, non-cholinergic pathway, activation of which releases nitric oxide, or VIP, to cause a gastric relaxation. So we have dual control over the stomach, which provides a very fine degree of modulation. These DMV neurons are my favourite neurons. They are tonically active. They are pacemaker neurons, and they fire at about 1 Hz, either in vivo or in vitro. So they are tonically active and providing a tonic input to the stomach. Predominantly a tonic cholinergic input. This pathway appears to be the more active one tonically. So the stomach is always under an excitatory control from the vagus. These neurons, however, receive all of this synaptic information. And because they sit, their resting membrane potential is within 1 or 2 millivolts of their firing threshold, it means that minor changes in their synaptic inputs can either shut those neurons off completely or radically increase their firing rate. This means that these neurons are exquisitely sensitive to synaptic inputs, so we can have rapid and finely tuned control of the stomach. Now, these vagal reflexes can occur independently. We can decerebrate rodents proximal to this level, and they will contain and will retain vagal reflex control of their peripheral organs to a certain degree. So that, however, ignores this vast connectivity of the NTS. And we know that the NTS receives both these parasympathetic innervation, but it also is one of the only areas in the CNS that also receives sympathetic innervation too. And the NTS is reciprocally connected to a huge number of midbrain and higher nuclei. And from a GI standpoint, what virtually all of these nuclei have in common is that they respond to stress, to anxiety, to emotional state. These areas are basically the autonomic output of the emotional state. So when you consider that, it's hardly surprising that stress and anxiety and emotions have such a profound effect on gastric functions because of this extreme connectivity through particularly the NTS. So while it's commonly said that what happens in vagus stays in vagus, and that's fine, what we in the Brain Gut Lab have been trying to convince everyone that we need to adopt the slogans, what happens in vagus gets relayed to the rest of the brain within 1 to 3 synapses. We admit that it's not particularly snappy, but we're hoping for the sake of accuracy it catches on. So what I would like to look now at is some of these other roles in physiology of the vagus. And the cholinergic anti-inflammatory reflex has been really important within the past several years and received a lot of attention. And there's a lot of excellent reviews being written about this cholinergic anti-inflammatory reflex, including this one from Kevin Tracy's group. So, as defined, pro-inflammatory cytokines and immune cell mediators activate the afferent vagus, which then relays this signal centrally. It's also important at this point to remember that this area of the brainstem, the NTS, the DMV, and the area postrema, is essentially a circumventricular organ. There are fenestrated capillaries, there's a leaky blood-brain barrier, and we know that circulating factors, including inflammatory mediators and immune cells, can activate those neurocircuits directly. But then as a result of this immune activation, we see a reflex activation of the efferent vagus to various organs, the liver, the GI tract, the heart, and also to the spleen, which decreases the production of pro-inflammatory cytokines. And as described over here, the efferent vagus is innervating the celiac ganglion, which is then causing activation of T cells within the spleen through a mechanism involving the release of noradrenaline, which then causes release of acetylcholine from the T cells, and this is decreasing pro-inflammatory cytokine production via a mechanism involving alpha-7 nicotinic acetylcholine receptor. Now, what was always a problem for people within vagal neurocircuit field was this idea that the efferent vagus innervated the celiac ganglion. Because what we would have then is a parasympathetic nerve innervating a sympathetic organ. And this was a problem that we couldn't seem to resolve. Now, certainly the early dye studies from Hans-Rudi Berthoud and Terry Powley, where they put neuronal tracers into the DMV, saw what looked like nerve terminals within the left celiac ganglion. So this suggested that there was actually a vagal input to the celiac ganglion. However, the anatomical studies never demonstrated whether or not these were fibers of passage or whether they were actually synaptic contacts. And doubt was thrown upon this idea of vagal innervation of the spleen or the celiac ganglion in this study from Pat Card's group, who used the pseudorabies virus. We injected this attenuated strain of the pseudorabies virus into the spleen. That's taken up and transported retrogradely, jumping active synapses. So it's a way of mapping connections within the brain. And they saw this pseudorabies virus in sympathetic areas of the brain. It might be hard to see, but we have the RVLM, the rostroventrilateral medulla here. We have parapyramidal neurons here. We have the NTS. But importantly, we do not have the DMV. And if the vagus innervated the spleen, we would expect to see DMV neurons lighting up. So this suggested that the vagus didn't innervate the spleen. And this went on for several years until finally Robin McCallan's group in Australia did functional studies. What they did was they would record splenic nerve activity and stimulate the vagus. And in doing so, stimulating the vagus, they actually did see an increase in splenic nerve activity. This is the average rectified integrated nerve activity. So stimulate the vagus, and they did see an increase in splenic nerve activity. However, they didn't find any stimulus-locked activation of splenic nerve activity, which you would expect if there was a synaptic connection there. And then they took the experiment even further and found that if they cut or crushed the vagus proximal to the site of stimulation, they lost the splenic nerve activity. So that implies that the vagus is increasing splenic nerve activity through an afferent pathway. It's a reflex response following activation of the afferent or sensory vagus. And so Patrice Guillenet's lab at the University of Virginia took this further. And they were looking at a model of kidney ischemia reperfusion injury. They would clip the renal artery for a period of time, causing an ischemia to the kidney, then release the clip, which would cause this reperfusion injury. And they can measure this ischemia reperfusion injury in many ways, in this instance, measuring plasma creatinine levels, which is a very good surrogate marker of glomerular filtration. And what they showed is that vagal nerve stimulation, if they stimulated the left vagus, they certainly attenuated the kidney ischemia reperfusion injury. They decreased plasma TNF-alpha, which would be expected from a cholinergic vagal anti-inflammatory pathway. It was abolished in alpha-7 nicotinic acetylcholine knockout mice, so that was good. And it was abolished in mice that had a prior splenectomy. Again, this would be good. And in fact, they confirmed the role of the spleen in a beautifully elegant way because they showed conditional transfer. So what they could do is stimulate the vagus nerve, collect the activated splenocytes from a donor mouse, transfer them into a recipient mouse, and that was sufficient to protect that recipient mouse from ischemia reperfusion injury. However, if they then sectioned the vagus nerve, they lost the protective actions of vagal nerve stimulation if they stimulated the efferent vagus, but they kept it if they stimulated the afferent. So again, it suggests it's a reflex response following activation of the afferent vagus. And just in case it was a possibility that the afferent vagus was activating the contralateral efferent vagus, they would section that as well. And they still got protection. So this suggests that the afferent vagus is activating some other pathway, most likely a sympathetic pathway, to protect the kidney. So what could this be? And in looking at the sympathetic nervous system, it's hardly surprising that that would be activated following these kinds of conditions because the sympathetic nervous system responds to physiological and psychological stressors. And the C1 neurons in the ventral medulla are particularly important in coordinating these responses. So the C1 neurons, as their name suggests, are adrenergic neurons. It's actually a very extensive nucleus in the rostrocaudal extent of the medulla. And from our point of view, it's very well connected to several areas, including the NTS and the DMV. And physiologically, these C1 neurons are involved in glucose and cardiovascular homeostasis. That's their job. But we also know that they are activated by all of these internal and external threats. C1 neurons are critically important in coordinating the body's response to hypoglycemia, infection and inflammation, hypoxia, nociception, hypertension, hemorrhage. In fact, this has prompted Patrice Guéhenné to coin these neurons, to term them as the body's EMTs, the body's emergency medical technicians. So are these C1 neurons involved in this cholinergic anti-inflammatory response? And he did this in a beautifully elegant study that was published earlier this year. So what he did was he transfected channel rhodopsin, so this light-activated ion channel, which will excite neurons. He transfected them under the tyrosine hydroxylase promoter into the C1 area neurons. So only the adrenergic neurons in the C1 area would express this channel rhodopsin. And then he could optogenetically activate these neurons 24 hours before he did the kidney ischemia reperfusion injury, and that was enough to protect the kidney from this ischemia reperfusion injury. And what I'm not showing here is he did the corollary experiment where he inhibited the C1 neurons and showed that inhibition of the C1 neurons attenuated the beneficial effects of vagal nerve stimulation. So the C1 neurons are sufficient and necessary to protect against ischemia reperfusion injury. And to further break my heart, and so that they showed that the vagus is not important in this, because if he did a subdiaphragmatic vagotomy, stimulated the C1 neurons, they still saw protection. So it appears that whatever vagal nerve stimulation is doing is an afferent activation of an efferent sympathetic limb. What does this mean in terms of the cholinergic anti-inflammatory reflex in the GI tract, which is a very dense vagal innervation? And certainly this study, again from Terry Powley, showed that macrophages are in very close proximity to myenteric neurons. But importantly, they do not receive direct innervation from the efferent vagus. However, studies from Guy Bockstein's lab showed that macrophages could be activated following vagal efferent nerve stimulation in a manner which involved the myenteric neuron. So we have the potential that vagal efferent outflow may cause activation of macrophages within the intestine. So what we think is happening is that if there is a local GI inflammation or immune challenge, we may activate an entirely vagally dependent cholinergic anti-inflammatory pathway. That means it's both afferent and efferent vagus. If, however, inflammation or immune challenge goes systemic, then it's more than likely that the majority of the cholinergic anti-inflammatory pathway is going to be through an efferent sympathetic loop. So, to switch gears then, does the vagus have any role in neurological disorders? Because we have heard several talks yesterday highlighting the non-motor symptoms which are pre-symptomatic in Parkinson's disease. And Parkinson's disease patients show a variety of these non-motor symptoms, including GI dysfunctions, which are present almost throughout the GI tract. And importantly, these GI dysfunctions are prodromal to the motor symptoms, in some cases decades before Parkinson's disease develops. And as Michel Neunlist explained yesterday, the potential involvement of the vagus nerve was highlighted in a very large retrospective study in Scandinavia, which showed that patients which underwent truncal vagotomy as a means of treating intractable ulcers had a significantly decreased incidence of going on to develop Parkinson's disease. So this led Brack to hypothesize this scheme for idiopathic Parkinson's disease etiology, where there was entry of a pathogen, some unknown pathogen, into the GI tract which infiltrated the enteric nervous system, causes misfolding of alpha-synuclein, and then there's retrograde transport either of that pathogen or of the pathological alpha-synuclein through vagal pathways eventually to reach the dorsal motor nucleus of the vagus, and thereon up to higher areas. The question was, how does it get from the DMV to the substantia nigra pars compacta? So, studies by my colleague Alberto Travagli investigated whether or not there's a direct pathway. And he did this first looking at, was there a monosynaptic connection here? And so he did this in two ways. He injected a retrograde neuronal tracer into the dorsal vagal complex. This would be uptaken by nerve terminals and then transported back to cell bodies. So you go to look, are there any labelled cell bodies in the substantia nigra pars compacta? And what you can see here, this is the retrograde tracer. This is the substantia nigra pars compacta stained for tyrosine hydroxylase, and it's kind of difficult with this light to see, but the arrows point to neurons which co-localize the neuronal tracer, suggesting that there are cell bodies that project down to the dorsal vagal complex. Then they did the converse experiment and injected an anterergo tracer into the substantia nigra. That will be taken up by the cell bodies and transported to the nerve terminals. Do we see nerve terminals in the dorsal vagal complex? This is the dorsal motor nucleus of the vagus here, stained for acetylcholine. And these higher magnification areas, we can see nerve terminals, which are contacting cholinergic vagal neurons, as well as neurons, tyrosine hydroxylase neurons of the A2 area. So there appears to be, these dyes do not jump synapses. So there appears to be a monosynaptic connection. Is it functional? Well, so they activated substantia nigra neurons by injecting NMDA, the glutamate agonist, and they see an increase in CFOS in the DMV and in the A2 area, but importantly not in other nearby nuclei. They didn't see CFOS either in the nucleus ambiguus or the hypoglossal nucleus or in the A1C1 region. So it appears to be a selective connection. It's not a generalized brainstem input from the substantia nigra. It appears to be selectively towards the dorsal vagal complex. Does it have any role to play in gastric motility? So here they applied miniaturized strain gauges to the stomach of anesthetized rodents and then microinjected NMDA into the substantia nigra. And we can see a significant increase in gastric motility and tone. That's abolished if you degenerate the substantia nigra with 6-hydroxydopamine. And it's vagally dependent because it's abolished following vagotomy. However, if I was being a really cruel reviewer, I would say, well, this doesn't prove it's a direct pathway because the substantia nigra projects to a great many other areas which may then impinge upon the DVC. So to prove it's a direct pathway, they did some optogenetic experiments. So they injected halorodopsin, which is an inhibitory light-activated ion channel, into the substantia nigra pars compacta. This would transfect those neurons and then be transported down to the nerve terminals. And then they could activate that inhibitory ion channel by shining green light onto the nerve terminals in the dorsal vagal complex. And what they showed is that green light stimulation inhibits gastric motility and tone at rest, as well as inhibiting the activated pathway. So that suggests not only that there's a monosynaptic nigrovagal connection, but that it's tonically active. So substantia nigra pars compacta is actually tonically regulating gastric motility and tone. And what I didn't have time to show you here is, but they have preliminary evidence that this nigrovagal pathway is compromised in animal models of Parkinson's disease. And it's compromised before you see full development of the locomotor symptoms, which is paralleling what is seen in humans. So the final things I would like to talk about are these potential for developmental modulation of vagal neurocircuits. So vagal neurocircuits develop late. In rodents, sensory afferents appear around about embryonic day 14, motor neurons about embryonic day 15, By the time we get to embryonic day 18, we have this rudimentary vagal reflex. These were in rats. Rats are born about embryonic day 21. But we know there's profound remodeling centrally within the first 2 weeks of postnatal life. There's a significant degree of synaptic pruning, reorganization, remodeling. And the circuits, central vagal circuits, are not fully patterned until about postnatal day 22 to 28. This is after they are weaned. And descending inputs from the hypothalamus, central nucleus of the amygdala, bed nucleus, stria terminalis, and the visceral cortex don't even reach the dorsal vagal complex until after birth. Now, that's in rodents. Rodents are born early relative to humans, but even in humans, the vagal system is not completely functional at birth. It's only partially myelinated. We have this rapid increase in total number of myelinated fibers through gestation, the greatest increase from 30 to 32 weeks gestation onwards, right up until 6 months postpartum. This period of time in rodents, the first 2 weeks, corresponds to the last trimester in humans. So there's a parallel modulation there. And we know that early life experience is really important in shaping adult responsiveness. Repeated brief maternal separation. We know that enhances active maternal care, reduces anxiety-like phenotypes in adults. That's work from Linda Reinemann's lab. And she also showed that it actually increases the inputs, the synaptic inputs from the hypothalamus to the dorsal vagal complex. In contrast, early life adversity, so this is more prolonged maternal separation, as well as other means of having an early life adverse event, increases stress axis dysfunction in adulthood, especially in females. It induces visceral hyperalgesia. This is work from Beverly Greenwood van Meervelde's lab. But interestingly, at least to me, because of my interest in dietary modulation of these neurocircuits, we know that maternal overnutrition increases stress reactivity in adults. And there's a lot of evidence in both animal and human studies for this. So the perinatal period, so the gestation and the first 4 weeks of lactation, is critically important in the development of these neurocircuits responsible for energy homeostasis. And both epidemiological and animal studies have said that maternal nutrition during this period is altering the predisposition of offspring to develop obesity in later life, and it's also modulating their stress responsiveness. We know it modulates hypothalamic neurocircuitry, but my question was, does it also alter vagal neurocircuitry? And to do this, we had to step back and have a painstaking study to figure out how these neurocircuits develop normally. If you remember, I said the most important synapse is this GABA one. Now, at birth, that's the first synapse that develops. But at birth, it is both GABA and glycine that are contained within those and released from those neurons. And it's actually excitatory to begin with until the chloride transporter becomes developed and reestablishes an adult chloride reversal potential. Both GABA and glycine are excitatory, and that's important for anchoring the synapse. Then the glycine currents decline, and by the third postnatal week of life, by P15 onwards, glycine is virtually absent, and GABA is the only neurotransmitter present. However, what we found is if we put animals, expose them to a perinatal high-fat diet, in adult control animals, strychnine, which is a glycine antagonist, does absolutely nothing to these inhibitory synaptic currents. In perinatal high-fat diet animals, however, strychnine is clearly having an effect here. It's increasing the duration and the decay of these currents, and that's because it is inhibiting fast glycine currents, and what we have left in red is slightly slower GABA currents. But the fact that strychnine is doing anything at all implies that both glycine and GABA currents are present in adulthood in rats that were exposed to a high-fat diet during the perinatal period. Does that have any role in excitability? Well, yes, it does. These are current clamp recordings looking at action potential firing rates, and as I remember, I said these were pacemaker neurons. And control animals show no response to strychnine at all, but perinatal high-fat diet animals are excited by strychnine because we are blocking an inhibitory input. So this implies that there's tonic activation of these glycine receptors in adulthood. Same thing happens in vivo if we do extracellular recordings from a whole animal, a whole anesthetized animal. Strychnine has no effect on the firing rate of DMV neurons in control animals, but it's increasing the firing rate in perinatal high-fat diet animals, again mirroring what we see in vitro. Does it have any effect on motility? Yes, it does. Again, we're applying miniature strain gauges to the corpus and antrum of these rats, micro-injecting strychnine into the dorsal motor nucleus of the vagus. No effect on gastric tone or motility in control animals, but a significant increase in both tone and motility in our perinatal high-fat diet animals. Even the highest dose of strychnine has no effect in our control animals. What's the important period of time here? If we measure the ability of strychnine to increase these inhibitory currents, because it's blocking the fast glycine, leaving the slower GABA ones. Control animals, no effect. Perinatal high-fat diet, we're seeing this increase. This is when dams were put on a perinatal high-fat diet from embryonic day 13 onwards, when the sensory afferents first appear, all the way through adulthood, and we're retaining glycine currents into adulthood. We know the perinatal period is important because if you put rats onto a high-fat diet at weaning, we're not uncovering a glycine current. And if they're on a high-fat diet and then we put them onto a control diet at weaning, we're not losing this, so it's permanently patterned. It looks like the lactation period is important. The embryonic period doesn't appear to matter because if we cross-foster pups, so you have 2 litters of pups, one dam is fed a control diet, one dam is fed a high-fat diet, and then you switch the pups. If we put, pups onto a high-fat diet at P1, postnatal day 1, then we retain this glycine current. If they're put onto a control diet at P1, no effect. And in fact, we've got it now timed that exposure to a high-fat diet for the first 10 days of life is sufficient to retain these glycine currents into adulthood. They can be on a high-fat diet during the embryonic period, switch to a control diet for 10 days, and then on a high-fat diet for the rest of their life, and we don't ever uncover this glycine current. So P1 to P10 is critically important in this developmental period. So what we think then is that exposure to this high-fat diet during this perinatal period is actually arresting the normal development of this synapse. And it means that inhibitory neurocircuits are retaining what would be considered a fetal phenotype into adulthood. So this is meaning that our vagal efferent motor neurons are less excitable, efferent output is decreased, and we're actually decreasing the vagally dependent tone of upper GI functions. And I thought this was very important and very exciting. I like synaptic physiology. This was enough for me, but I understood when other people sort of looked sympathetically and shrugged their shoulders at me, and what does this mean anything more to it than this? And potentially, yes, although we haven't done the studies yet, because there may be a physiological and pathophysiological additional importance here, because glycine currents are sensitive to a whole array of allosteric modulators. Glycine currents are exquisitely sensitive to alcohol and anesthetics. What that would suggest then, I would expect these rodents to have exaggerated vagal inhibition following exposure to alcohol or anesthetics, neurosteroids as well, including sex hormones, which may have quite profound implications for females in terms of vagal control of GI functions, And also glucose and insulin are positive allosteric modulators of a glycine channel. These rats are predisposed to put on weight, to increase feeding, to develop metabolic syndrome and type 2 diabetes. And as their glucose levels and insulin levels begin to rise, we are going to exaggerate this vagally dependent inhibition, which is actually going to exaggerate their food intake because of this inhibition of gastric functions. Excuse me. So, what I've hoped is that I've convinced you is that vagal neurocircuits are not static. They are not unmodulated input-output relay systems. They're actually modulated by a variety of homeostatic influence, feeding, fasting, time of the day, stress, circadian rhythms. Vagal neurocircuits, particularly the NTS, it's a critical link between the sympathetic and parasympathetic systems. It is the only area in the brainstem that receives both sympathetic and parasympathetic inputs. Central vagal neurocircuits are highly interconnected to a whole array of other brainstem and higher CNS centres, and that may be central to GI dysfunctions and neurological disorders. And finally, the functionality and responsiveness of vagal neurocircuits in adulthood may be dictated by maternal and environmental influences during very critical perinatal developmental time periods. So, just remains to thank everyone in the Brain Gut Lab. The Parkinson's disease work are studies which hopefully will be published soon by Alberto Travagli, and as anybody who knows him will know that while he was off fishing, all of the work was being done by Laura Anselmi and Cecilia Bove. The developmental study, which I talked about at the end, was spearheaded almost entirely by my student who made some very observant observations, Caitlin McMenamin, who's since graduated and is now at Rutgers University, and also by Courtney Clyburn. Special thanks to Sean Stalker, a collaborator at University of Pittsburgh, who did all of the extracellular and juxtacellular recordings, and particularly to Holly Travagli, our lab manager, without whom everything would collapse within about 3 days, and the sources of funding. Thank you.